RAI.
Yeah, RAI.
Yeah.
All right. Okay. Thank you.
Yeah, thank you, Professor Rai, for an exciting talk. I think I will just let the organizer to take over, because I think the other speaker have not joined yet. Still time is there. So over to the organizer, please.
Thank you. Thank you very much.
Yeah, probably the next speaker is me, Dr. Rajiv from IIT Guwahati.
Yeah. It's a good morning. Yeah. So let me just introduce you. This, we are very glad that Professor Rajiv K is and a PhD degree in ISC Bangalore, and his research interest in the areas of biological devices, spectroscopy, nanotechnology, and quantum protein simulations. So I'm very thankful that he is here. So please, Professor Kar, you can just share your presentation and then start your presentation. Thank you.
Yeah, thank you very much. And I just need the permission to share the screen.
Yeah, I don't know. The organizers can tell. Actually, I am not sure. You can just try.
Oh, okay. We are giving the permission.
Yeah. Please do that. Thank you.
Okay. Now, sir, you can try.
Yeah. So, yeah, I shared the full screen, and now possibly everyone can see my slides.
I can see. You can put the full screen. Yes.
Yeah. Thank you very much. I'll just use the laser pointer. Yeah. So thank you very much for this wonderful introduction. And also, I would like to thank Professor Panigrahi and Professor Sen for giving me this opportunity to speak on this platform. And today I'll be speaking on this tuning the quantum chemical properties of biological chromophore. So unlike the other presentations where we talk in details about the algorithms, the method development, and the main development features of quantum mechanics. So this talk will be focusing on the application part. And most probably we will be talking about the chromophores that are present in biological scenario. And we will see how the interface of quantum mechanics and the spectroscopy are helpful in understanding the overall phenomena. So before going into depth of the presentation, this will be the outline. So I'll be first talking about the very basics, starting from scratch to make the audience aware of the topic. And then we will be talking about the differential role. In principle, we will be discussing about the classical mechanics and the difference of the quantum mechanics. So herein we will discuss what kind of problems that are possible to solve through the classical mechanics, and in more precise form, why we basically need to solve to use quantum mechanics in order to solve some complex problems. And then we will also discuss a new possibility where we can account for the hybridization of both the methods to understand the phenomena, be it biological or be it chemical in origin, in more details. And lastly, we will see the applications through two typical examples. So where we will have this flavin and retinal as biological chromophore. So as you can see, both of them are highly conjugated system, and that's why they are much more suitable to study through the quantum mechanics. So now, as I said, like we will be talking about the briefs first. So the first slide is about the purpose of molecular modeling, because so far the quantum mechanics principle that we want to use is through the computational methods. And here we actually work on models. So more precisely the toy models that represent the real behavior of universe. So the purpose of these molecular modeling is, number one, to create models where we can see the network, the three-dimensional arrangement. So for example, this is a model of protein. And of course, it is useful for the visualization part. So here we see that there are different color codes, and this represent different proteins that are interacting with each other, and the statistical insights that we will gain through the simulations. But more importantly, the reason we use the the molecular modeling principles is to assign the experimental measurements. That's where it is of more importance, because in circumstances where the experimentalist can do the measurements, but they are skeptical of what is going behind the peak and what are the reasons, there comes the importance of molecular modeling, where we can pinpoint the use and the relevance of each atoms, and more precisely each electrons in governing the overall phenomena. So for example, we want to understand the mechanism of experimental outcome, or the interaction details, be it atomistic or electronic in nature, and also for quantifying several measurement peaks. So for example, like the particle size, wavelength, and energy things. So now, as I said, like once we are done with the basics of molecular modeling, the next principle is to use the spectroscopic methods. So in order to explain the spectroscopic phenomena, basically we can either use classical mechanics or quantum mechanics. But then the main issue is we can solve the macroscopic features with the classical, but not the very tiny details which are arising from the electronic origin. So from the physical theory, which accurately describes the behavior of atomistic properties, the quantum mechanics is of high relevance. And in order to do so, as I said, like scientists are basically generating models to predict the universal behavior. And here we are, rather than focusing on the whole system, we are focusing on a very tiny and small part of this system. So we will see in details of these. So now, this is something again, which we need just to introduce the audience. So for example, we have a lot of tiny spheres. So anyone can answer from the audience, like what we can, depict out of this picture. Anyone? Yeah, I'll be happy to listen, like in case any guess is there. All right, so if everyone is still guessing, so these are tiny spheres, but then in order to distinguish them from each other, what we can do is like we can account for their van der Waals radii. And this is like the atomic radii, which can be the first point of distinguishing one sphere from another. And then we can actually refer them to the element like hydrogen, nitrogen, oxygen, and other heavy atoms. So this is one criteria where we can train the computers to understand like how we can represent the different atoms. And the next thing is about the charges through which we can describe them. For example, when we have this famous Rutherford atomic model, we see that there are neutrons and protons, but then on the outer shell, we have electrons. Now, these electrons are playing a major role in order to describe the valence shell and also to understand the electronic properties. Now, this is the typical representation for every atom. And when we talk about the bonding between different atoms, say, for example, in water or in carbon dioxide and other molecules, so this is like the sharing of the electrons through which the bond phenomena is visible to us. Now, here the important aspect is the partial charges or the formal charges. So be it the bonding phenomena where the charges is being distributed among the atoms, so it will be the partial. And in the whole set, or for example, in the ionic cases, we have the formal charges where we depict +1 or -1 and so on and so forth. So these are the two basic categories where we define the parameters, be it the van der Waals radii or the charges. And now we will discuss about the classical mechanics. So in principle, the classical mechanics used to describe the macroscopic features, and this will be describing in terms of either non-bonded interaction, which will be through space, or it will be bonded, which is like via connection of more than two atoms through bonds. So when we have the non-bonded terms, we have this Lena-Jones potential or the van der Waals kind of interactions, and we also have the Coulomb or the electrostatic interactions. So they are being denoted by the two equations. And then we have the bonded terms. So we have this for two atom system, or for three where we have the angle, or for four where we have torsion or dihedrals to describe the plane. And taking together, these parameters are useful to describe the system as a whole. And since classical mechanics is a bit of crude, so it is possible to account for, say, thousands of atoms and to describe the macroscopic behavior. Now, these are some of the insights that are possible through the classical treatment. Say, for example, we want to look at the energy landscape. So we can do some simulations, and then we can quantify the conformations, and then we can trace the rate of kinetics between transition between one state to another, and this is how the energy landscape can be described. And likewise, we can also account for the secondary structure where the alpha helix, beta sheets, or random coil turns can be described. And as I discussed the partial and formal charges concept, so the surface phenomena of or the electrostatic interaction through which the protein-protein interaction happens, that can also be described, and this comes within the classical treatment. And other than this, the interaction energy, be it protein-protein or protein small molecule docking, or in the major conformational states of membrane, we can also see the dynamic stability. So these are some of the things where we can apply the classical treatment, but it will give the overall behavior and not the precise information. So rather than quantification, I'll say these are the qualitative terms that are possible through the classical treatment. And if we talk about the spectroscopic features, so sometimes we see dynamic light scattering or some like circular dichroism. So these are, although they are also working with the electromagnetic radiation, but they corresponds to the classical mechanics, which can be elucidated using this. But then when we want to model the electronic properties, there comes the bottleneck, and it is not possible to apply the simple classical mechanics there and elucidate the property. So for example, when we want to see something which is called bond breaking or bond formation, so this is one example where there is a biological chromophore and there is some chemical moieties, and then later on, in presence of light with the external photons, so there is some bond formation, and this is something more complex which is not possible to study through the classical mechanics. So likewise, we can also account for bond isomerization, although it looks very simple, but then it gives rise to major conformational changes and several signaling cascades within the biological system. And so they are also well defined through the bond isomer through the quantum mechanical principles. And likewise, we can also account for the orbitals involved in the ground states and the excited state transition, and also we can predict or determine the spectroscopic properties. So these are in a nutshell, like the properties and the features that can be elucidated using the so-called quantum mechanics, and where they are much more easier. So in principle, if you want to compare the classical and quantum phenomena, classical looks much simpler. It's just riding a slope. So the bigger the system, it will be a little bit costly, but then still we are able to calculate. But then when we come to quantum, it's more complex, and by principle we know that no two electrons can have the same quantum numbers, and so there comes a little bit complexities. So if we look at the physics behind, so based, as I said, it can account for the macroscopic features. And so if we want to approximate the physical quantities, it is possible where we can account for bigger system. But then when we want to have highly accurate and reliable model, we definitely need quantum mechanics. And this is the main reason, like when we try to come to the potential energy minima through the classical mechanics, we will somehow be far away from the realistic model, which can be obtained from the quantum mechanics. So there will be a significant shift in terms of energetics, and that's why this is more reliable. So now the challenge, as I said, it's challenge is mainly lying in terms of computational expensiveness, and that's why we cannot account for larger system. And say, for example, a protein which comprises thousands of atoms, so it's very difficult to solve these wave functions and the operators. But then, thankful to these three gentlemen who came with a modified concept, and because of that, they won the Nobel Prize in the year 2013 for the so-called development of multi-scale models for complex systems. So let's understand the principle of this hybrid quantum mechanics and molecular mechanics. To illustrate, I took this figure from Nobel Prize website, where we see the picture of Alfred Nobel. So you can see this innermost region, which is this bounded by this white line. So this is very highly resoluted. So you can clearly distinguish every tiny detail of the phase. But as soon as we come out of this core, we can see the next layer is bit pixelated. Although we can see where is coat, shirt, and tie, but we cannot precisely define the borders. And this is something like where with minimal efforts, we can actually describe the system. And then comes the third layer, where we cannot understand what it is, but we can presume that there is some environment existing. So this is the outermost layer. And with this, we can actually reduce the complexity, and also we can reduce the computation overload, and then we can be able to solve the complex problems lying within the biochemical system. So by doing so, here is one example, like where we can see the experimental or the model structure, which is shown in the black. So this is the potential energy minima. But when we apply to the the molecular mechanics, it can solve some of the problems associated with the frozen structure. But then we will again be stuck at just a near minima to that. But when we apply this quantum mechanics and molecular mechanics principle, we can get into far more realistic energetic level, and it will describe in more accurate terms. So here also the important lies, because we can not only account for the ground state, but also the excited state, and then the difference between the minimas to the next excited level, and coming back, we can actually measure we can actually calculate the absorbance or the fluorescence behavior. So this energetics basically corresponds to the spectroscopic measurements, and then we can actually make a synergy between the analysis. So this is the schematic representation where we understand the hybrid hybrid quantum mechanics and molecular mechanics principle. Now, now I will describe some of the methods that is, applicable to the biological system, because although we see there are a lot of complex systems, but then not a single method is applicable to every system. And that's because sometimes our insights or the objective of understanding is different. And sometimes we want to, corroborate the complex things with the spectroscopic measurements. So this is just the most simplest form. So here you can see a chromophore, and rather than describing the near environment, say for example the water molecules or the protein environment, we simply describe some environment, and that description of environment is actually denoted by this epsilon, which is the dielectric of this medium. So for example, this one I describe with water, and we use this dielectric constant to suggest that there is an implicit environment, and we are not accounting for individual atom-based properties, but the overall effect. And this will give us, the simpler quantum mechanical effect on the system. Now, to be a little bit more complex, we need not to account for the full major conformational state of the protein, but then we can only extract the near environment, that is, say, the first shell, which comprises of, say, 4 to 5 Armstrong distance from the chromophore. And that part we can actually extract from the whole system, and we can study it. So although this will be a little bit expensive, but then this is much accurate compared to the implicit medium conditions. So here we describe the atomistic model of the nearby environment, say the protein, but then not fully, only to the part which is in the close vicinity of the biological chromophore. So now the perturbative effect of the environment will be much more rigorous. The next case is we want to apply some more complex the higher level method, but then again we cannot afford more than say 100 atoms at a time. In that scenario, what we usually do is we take account of individual partners. So for example, the chromophore with one or two amino acids or the solvent molecules, which is very close to it. So they have some through space interaction, but then the electronic correlation can be accounted using a bit higher method. And this is known as fragment molecular orbital method, where we take account of the chromophore, but then also one or two residues, which is in the close periphery. So this is another method of application of quantum mechanics, molecular mechanics in the hybrid form. And now we come to the mainframe, where we are accounting for the full system. And this is possible by treating the system with different resolutions. So as I described in the previous slide, so the mainframe is the chromophore, which is central to the system. And we believe like major electronic transitions are being governed by this chromophore. And the potential energy surface of the chromophore is basically modulated by the environment. So that is the reason the reaction center, which is the main area of focus, is being described by the quantum mechanics. And to the outer, we have this molecular mechanics part. So this MM basically describes the whole protein, but QM describes the major area, that is the chromophore. And in certain cases where we want to see what will be the the effect of the near environment with a more rigorous method rather than the whole system, we can also apply different resolution levels. So for example, the center of interest, that is the chromophore, it have the say described by one quantum mechanical level, and then we describe by another quantum mechanical level to the amino acids, which is in close proximity. So these two, QM1 and QM2, basically corresponds to this kind of cluster system. And then the remainder of the system will be treated as such with the molecular mechanics. Now, the essence of this molecular mechanics is it will not be describing everything as a whole, but rather the more concern is about the point charges, or we can say the partial charges. So because each of the heavy atoms present, they will be being affected by their environment, and then ultimately this will be a perturbative effect on the subsequent code. So that's why the dynamics that will be governed by the molecular mechanics will be compensated in terms of the partial charges. So be it like the negative or be it the positive. And then this perturbative effect will be accounted through the quantum mechanics region. So these are some of the methods with which we can actually account for the biological system, and we can describe the complex phenomena. And we can basically, corroborate with the spectroscopic features. So now let's see some applications of this quantum mechanics. So as of now, like I think many people are well aware of time-dependent density function theory, but then from the application point of view, we can say this is as a black box method. The reason being for such practice, we need to describe the whole system in terms of the coordinates, and then we also need to describe the functionals and the basis set, and that's it. And rest the algorithm will do for us. And what it does is basically it will account for the highest occupied molecular orbitals, or the and also the lowest unoccupied molecular orbitals, where the electrons from the ground state will be transitioning in the excited state. So this is as such, as I said, we are not accounting for the individual molecular orbitals, like the linear combinations, we are not accounting as a whole. So that is why the TDDFT and other methods could be accounted as black box method. On the other hand, if we want to be bit more expensive, but rather than this, we are aiming to achieve more reliable phenomena, then we have some advanced features. So for example, this is called active space selection of molecular orbitals, where we can account for the complete active space perturbative methods or n electron valence electrons. So there we can actually take control of the molecular orbitals involved in the transition. Say, for example, the lone pair on, say, oxygen, we know that this orbital is not involved. We can actually exclude, rather we can include some other lower energy HOMO orbitals here. So these are some methods where we need much deeper chemical intuitions in order to govern the calculations. So that's why they are not black box, but rather some advanced methods. And as I noted, the title of this particular slide is to assign experimental spectra. So here is an example where we are trying to assign the experimentally measured Raman shifts. So in the bottom line, you can see the experimentally measured Raman peak and resonance Raman peak of the same flavin molecule. And on top of it is the calculated one. So ideally, you can see there are some shifts, and that's because not every physical phenomena can be accounted into this quantum mechanical calculations. Rather, we are also accounting a lot of approximations, and that gives some discrimination. I mean, there will be a bit of shift, but then we can assign in order to understand what's going on in the experimental spectrum. So from this, we are inferred that the pattern, overall trend that is observed in the experimental one is being reproduced in the calculations. And so we can now take into look of the atomistic models where we will find the normal modes associated with each of the frequency peak at different location. And then we can go back to our experimental one and we can assign one by one. And this is how the fruitfulness or the usefulness of this quantum mechanical calculations come into practice, where we are not just going as a black box, but rather we are using these predictions in order to assign the experimental measurements. So likewise, I'll give you some more examples. So here we are also accounting for the same flavin molecule, but then the context is a bit different. So here you can see two NMR tubes where we have the same sample, but then the color is different. And the reason for being the color difference in color is because now the molecule, although it was same in the starting state, but now it is a little bit different. So meaning they like one of them got earlier it was yellow, but then because of some reduction, some oxidation process, it becomes red. So this infers that although we can see the classical phenomena, but then these things happens very quickly. And then it is quite difficult for the experimentalists sometimes to capture. So as for example, you can see when the sample was prepared and immediately we do the measurement, it was blue. But then, with some time variation, if we allow it to incubate for some time, it will undergo some oxidation process, and then the redox state of the molecule completely goes into a different one. And that gives a different characteristics again. So although UV is a very well-known and reliable method for characterization, but it suggests that the changes that happens in the redox state of the molecule is quite fast. And if we allow for some time, we see a complete different pattern. Now, from the experimental point of view, it is very difficult to, capture these states and then to look into the tiny details. But rather, if we apply the quantum mechanical principles, we are now in a very good state to understand what's going on at the molecular level, and then we can assign the reason. So for example, here we have the completely oxidized state of the flavin molecule. But then when it absorbs a electron, or it absorbs electron along with some proton, it will go into either a semi-covalent state or neutral semi-covalent state. Again, in the next state, when it will again accept one more electron or a proton, then it will become a hydroquinone state, or again, after getting one more proton, it will become neutral hydroquinone. So these are some of the states. Again, they are interchangeable, but then it depends on the conditions that is being presented over there. So ideally, we can see that there are different states, but then the kinetics between kinetics for the transition between one to another will be very fast. And so rather than completely relying on the experimental measurements, it's quite effective if we apply the quantum mechanics and understand the tiny details of the phenomena. So what we inferred from our calculations and also from the spectroscopic measurements, that basically this is being held at this particular diazobutadiene fragment of the entire molecule, because this is quite a reactive part, and all the electron transfer and the proton transfer basically is mediated through this 1, 10A, 4A, and N5 this bond. And also, although the flavine is quite planar in structure, but then upon accepting the electron or the proton, it undergoes some butterfly-like conformation, where this dihedral or this dihedral will be quite of high relevance. And as a result, there will be some change in the angle, and this is what we can basically understand through our computation and application of quantum mechanics, and then we can actually make inference to why these transitions are actually happening. And in addition to do this, like when we want to assign the individual transitions happening within the peak, and why sometimes the peak vanishes and why a new peak or the shifted peak arises, again the same phenomena of the molecular distortion as well as the electronic changes or the change in the electron cloud basically depicts them. So I hope I am able to convey my message, why we are using the quantum mechanics as such in order to predict the property, or in order to replicate the experimental phenomena, and then to understand from the molecular details like what is going on and why we are seeing the classical effects in a macroscopic view. So now I'll give you one more example of use of this quantum mechanics. So in this case, we took into account of this GPCR like protein. So because this is G protein complex, G coupled receptor, and there we have 7 transmembrane helices, and in between there is a biological chromophore. So if we look into details, so this is the biological chromophore, and it is being supported by some hydrogen bonding through some amino acids and also some water molecules. So this is the typical photocycle where as soon as the protein is expressed, it is stable in the dark condition, and once it is being illuminated with light, there will be some activation, and with some delay gap of say picoseconds to nanoseconds, there will be different conformation where it will either accept an electron or a proton, and ultimately there will be some changes in the conformation, that is the isomeric changes, which will actually lead to difference in the overall conformation and the signaling cascade. So basically what I mean to say is, this is the chromophore which will play the main, the role of being at the reaction center. As soon as light falls on it, it undergoes some isomerization, which will lead to a major conformational change in the protein, and that allows a sodium ion to pass through. So this is a channel where the sodium ion can go, and then the cellular process can happen. Now, the problem that we faced is while capturing the time-resolved structure of this GPCR protein, it was well known that in the dark state, when the protein was expressed and it was in stable condition, there is a water molecule near this this retinal molecule. All right, but then then as soon as the light falls, there should be a sodium ion that will be passing on. And the tentative position of the sodium ion is quite challenging, because Na plus and water molecule, the oxygen, have the same kind of electron density. So it was not possible to identify whether in some illuminated state there will be water itself or there will be sodium ion. And of course, when we mutate the 2 amino acids which are at the close vicinity and responsible for the stability of the chromophore, the complete functioning of the protein is vanishing. So ideally, these 2 are important, but then this mainframe where water molecule or the sodium ion remains is a question. So this was in all trans conformation. Now this is in cis conformation where isomerization happened. But then upon isomerization, there will be conformational change and there should be space for sodium. So in order to do this, we first calculated the dark state with water and the light state with either water or the sodium ion. And this is the result. So the experimental spectra for the resting state, that is dark state, and the light state where we have the sodium ion have a shift of this 66 nanometer. And this was towards red, that is higher wavelength or lower energy. And then when we do the calculations, we found a similar trend. That is when we have the sodium ion, it was red shifted and it was in line with the experimental measurement. But as soon as we retain the water molecule instead of sodium ion, the excitation energy goes in the negative direction, and that clearly rules out obtained indeed have a sodium ion and not the water ion. So this is one of the beauty of quantum mechanics, where we can pinpoint the relevance of electron densities, and then we can also describe the biological phenomena. Now, why we basically want to study the isomerization? This is also a very important question. So the reason is like, we will come this to later, but then imagine the same biological chromophore is present in different protein, and all of them have a different kind of role. So for example, here using the quantum mechanics, we want to understand how the protein is actually governing the overall isomerization mechanism. So these are some cartoon illustrations where we account like different environment, and then how the protein environment tunes the potential energy surface of the chromophore, and how the isomerization actually happens. So it is not as such straightforward in terms of what we learn from the quantum, what we learn from the basic chemistry notebook, be it cis and trans, but the path of achieving these two states are quite different. And in order to give analogy with the real life system, so here is one model where many people suggest, like, bicycle pedal mechanism, where the two bonds associated near the point of isomerization will behave differently, and the cis to trans state will be achieved. Likewise, there will be this hula twist, which comes from the famous dance of North America. And it says, like, again, some bond which is associated just near to the main point of isomerization, they will be, making some twists. Or it could be bring a wet towel where, and this rotation will take place. Or it may be more than one bond will be actively, isomerizing, so as to give rise to the final state from trans to cis conditions. So these are some of the analogies that are being elucidated exclusively using the quantum mechanics, because as such, from the measurements, it's quite difficult to pinpoint these changes and to make some inference like how the mechanism is actually happening. Now, the next state is basically, as I said, like, why we are studying this. Now, this is quite important. So imagine this is like the retinal chromophore that was actually taken out of the protein environment. And when we put it in normal solution, we see that the color of the retinal is somehow close to yellow. And this suggests like it is somewhat lying around, say, 400 nanometer to 500 nanometer, something like this. But then as soon as nature put this molecule into different proteins, so this is a phylogenetic tree where we have different class of species which are linked with each other in terms of evolutionary relationship. And so some of them are sensory retinal, some of them are ACR or sodium pump or xenoreceptors like this, or channel rhodopsin. So we see that when nature put the molecule in different kind of protein, all of them tends to behave in a different way. That's because even though the molecule or the biological chromophore remains the same, but the near protein environment is different, which actually in terms to tune the potential energy surface of this chromophore. And that's why we see the excitation from the ground state to the excited state. I mean, the energy gap is now quite different. So when it is, very less and like lower in terms of energetics, we see something near the red. And when the energy gap between the ground state and excited state is quite high, we see it is near towards blue. And this difference now, as I said, is arising because of the near environment. So that means the amino acid, which is in the close proximity of these chromophores, they actually are either electron withdrawing or the electron donating kind of species. And so they will modulate the electron density of the chromophore, and ultimately the overall, the colorimetric phenomena will be changed. So I hope now it is quite clear, like although we can see a lot of differences from the colorimetric phenomena or the overall like visual inspection, but then the basic the basic driving force is the potential energy surface modulation, which is now accounted within the biological system through the amino acids. And then when we want to pinpoint how they actually correlate, that is actually being supported through the quantum mechanics. So in principle, we can understand the spectroscopic tuning, as I said, the excitation energy differences between all of these systems. And then with this, we can actually understand how we can develop the mutants for the medical application. So I'll give you an example. So everybody who can see my screen, rather than looking at the presentation, you can see my camera now. So as I can show you that this is a white light of my mobile camera. So now if I put white light in front of my fingers, you can see that some red color is coming out. So basically, this means that out of all the the visible spectra, only the red light is able to penetrate the skin and other like blood and other membranes. So this red light, being very low in terms of energy, is able to penetrate and transverse across the cells and tissues, and it will also cause minimum damage. So on the contrary, if we look at the blue light and others which have high energy, they can actually do some damage to the tissue if we use it for the medical purpose in a focused view. So this is the reason that we want to understand the nature of the absorbance or the features that are supporting these colors. And then the ultimate objective is to construct some artificial protein that will infer some red color of absorption. Because if it is near red, it can be then applied for the control of cellular dynamics using external light. And this field is actually known as optogenetics, where we want to engineer the protein, and we want to understand something which is near towards red light or the lower energy phenomena. So so this is the main importance of why we want to apply quantum mechanics in these kind of photo systems and understand the overall phenomena that is going on. Now, since I gave you one example where we see the difference in colors, and now I will give you the same thing in terms of, in a very basic form, that is reproducing the absorption spectra. So for that, instead of retinal, I'll come back again to my favorite molecule, that is flavin. And flavin as such, we know, like, it is quite common because we often take it as folate supplement, or in some countries it is also being used in breads and other consumables. So flavin is quite common in our day-to-day application. So now the flavin, although they have versatile role in biological system, but then they also have key importance in terms of designing sensors that can actually help us in controlling the cellular processes. So here is one example where we will study the flavin isoalloxazine moiety, but instead of this C8 position, like instead of having the traditional methyl group, we can also have different kind of groups available that is being learned from nature. Now, the importance is we want to again go to the synthetic biology, where we can control the absorption spectra and we can make it use in optogenetic phenomena. So I'll show some examples where we want to actually reproduce absorbance spectra using these quantum mechanical methods. So at first you can see these are two absorption spectra where in one we have the normal flavin, we have CS3, and another we have amine group. So these are two typical examples. And the spectrum in black is the absorbance. Here also the spectrum in black is the absorbance that is being collected through experimental measurements. All right. And now when we took one conformation of this molecule and we optimized it, and then we calculated the vertical excitation energies using the traditional TDDFT or ab initio methods. So now based on this vertical excitation energy, we then simulate the Gaussian spectra. And now these are two methods, namely the coupled cluster method or algebraic diagrammatic construct second order method, ADC2. So these two methods we try to now compare with the measured spectrum. So as you can see, there is a lot of discrepancy, although this low energy peak is falling very nicely to that of the experimental measurement, but then the second peak is quite shifted. Third peak, we are not aware because the experimental measurement was stopped here. On the other hand, for the amine group, we see again, there is a discrepancy for the lower energy peak because the Gaussian broadening is not matching, and the peak positioning is also a little bit shifted. But then this second peak is somehow not being reproduced in the calculations, and the third peak is not matching in terms of absorbance or the oscillatory strength that we get from the quantum mechanical applications. So when we want to look into the electron density difference, we see when we are on the lower energy state, that is S0 to S1, the first one. So we see a very clear distinction between, electron positive or electronegative densities because of the transition between the ground state and the excited state. So that's why these two peaks are well reproduced. But then in case of amine, for the second transition, there is no big, separation between the red and blue contours, and that is probably the reason this second peak is very much less in terms of intensity, and while in normal flavin it is having a very clear intensity. So now the question is, although we want to apply the quantum mechanics, but we can observe the rudimentary things that is very basic, that what kind of electronic transition being involved in this, but then we cannot reproduce the spectrum. So in order to do this, rather than taking just one conformation, it is now important to look at the ensemble, because when we look at the test tube, we are not remaining with just a single conformation. Rather, we are having an ensemble of conformations where we have different states, and all of them are contributing in some sense to the overall spectroscopic phenomenon. So in order to do this, we then calculated a long trajectory. We collected the snapshot, and now instead of relying on just one structural calculation, we did the same for say 50 states, like 50 different conformation or snapshots. And now you can see these are S1 to S10, that is the excitation states. Some of them are dark without the oscillator strength, and some of them are having the oscillator strength, which corresponds to the absorbance. So now we can see the Gaussian broadening is well reproduced, although there is a bit of shift, but then because of this Gaussian distribution, we confirm it is very close to the experimental structure. Also, we now reproduce the second transition that was failed in the single conformational state, and that's because some bending, butterfly kind of bending, or some angular the planar change happened in the geometry of isoellipsis ring, which was not accounting for this state earlier. But now, because we have several snapshots, so some of them actually corresponds to this. And then the high energy peak was very beautifully reproduced. So this is an example where, although in some cases we can account for the measurement, but then we can also now translate this technology or this method for the machine learning and AI-based tools, where we can simulate the spectrum for hundreds of molecules. So another thing is the role of protonation. So this was one condition where we can actually reproduce the absorbance spectra. And now I will look into some more complex phenomena where, instead of one, we have now two chromophores. That is the same flavin, but it is present in a different site at within a protein. So you can see when there is some NADH2, like some reaction is going on, 2 electrons are generated. But then the 2 electrons are being diverted to flavins separately. And how nature is governing this role is quite phenomenal, but we are not fully in a state to understand this. So if we see the chromophore, they are same, but at different sites they are being connected through different residues or the water molecules or some other environment. And that's basically governing the not only the potential energy surface, but also the state through which it will be interacting with the near environment. So in this work, we have a lot of complex system understanding, but then I'm just showing you one particular example where we look at the histidine protonation state. Because in histidine, we have 2 titrable nitrogens, and so the proton could be aligned to either epsilon nitrogen or it could be to delta nitrogen. So then we place the proton on either of the 2 state, and we try to elucidate the free energy with respect to the distance of interaction. So what we understood is this yellow corresponds to the state where histidine have hydrogen on the epsilon, and the cyan one corresponds to the other one, that is the proton is present on the delta state. So what we understood, so each tiny point is actually independent calculation, and then we plotted the free energy state. So what we see here is when it comes to the epsilon histidine, we have only one minimum, but then again it is not fully to the lower energy state. However, when we have the delta one, then we reach to the free energy state at a much lower level, and also there are more than one minima that suggests the conformational dynamicity also allows this state. So this is again one case where even when we want to go for the cryo-EM or X-ray diffraction methods, it's not possible to understand these phenomena, but rather the quantum mechanics is at the superior position where we understood what is going on and which one is more favorable in allowed form as permitted by the nature itself. So this is then, and now I'll go to just last example of the same flavin-based protein where we are looking for designing sensors. So as I said, one phenomena is like when we see the chromophore, it is although same, but then when it present in different proteins, it governs different phenomena. And that's because the modulation by the protein environment is actually phenomenal there. So this is one case example where we can use the LOV, that is light, oxygen, and voltage. So this stands for LOV, and this is the name of typical name of the flavoprotein where the active chromophore is the flavin, and the nearby environment plays a role. So as soon as there is the light and typically of wavelength for 450 nanometer, it will inject some, it will trigger basically the molecular changes. And so the nearby amino acids close to the chromophore will actually make some cascading changes. And as a result, we can actually govern the on or off phenomena. So we can actually modulate whether the changes should proceed in forward direction or it should be stopped immediately. So this is possible by basically understanding the protein environment, and then we can use some mutants to understand it. So this is the typical example of LOV protein. So the major domain is the one where we have the chromophore and the protein. Besides that, we have this helical region, which is closely connected, and there is another protein motif. So when light comes, this part will be activated. This helix will be uncaged. That is, it will undergo some conformational changes, and this effector part can be now movable. So we can basically control the light-induced effect here, which will then control other two phenomena. So what happens at the very molecular level is, this is a flavin, we have the protein, and there is a cysteine residue, which have this SH group, thiol group on the terminal of the amino acid side chain. So now when light of 450 nanometer falls, there will be a bond formation. As you can see here, there will be a bond formation, and also the proton, which was earlier present here, will now be shifting towards the nitrogen. And this tiny part, which is the bond formation and the proton movement, actually governs the major conformational role in the biological signaling cascade and overall phenomena happen. So imagine if we modulate now this process, again, as you see, there are two arrows, so that is, it is reversible. So when light is there, it will make a bond, it will stay for some time, and again it will come back to original state. So this delay is also, we can refer it as photocycle kinetics. And now, by understanding the near amino acid environment, we can actually tune this photocycle kinetic. So when we want something, it should be there for prolonged time. We can inject, mutate with some electron donating group, which will facilitate this bond formation, and so the photocycle kinetics can be extended. And when we do not want the cellular phenomena to happen, we will supplement with, say, some groups which are not at all donating the electrons, rather they are steric group. And so it will not facilitate this chemical reaction, and so the photocycle kinetic will be of short-lived states. So the application mainly lies in treatment of diabetes, heart disease, and even cancer. And it is still within the development regime, because we are yet to understand the full quantum mechanical phenomena that is happening, and then to translate it into the real application of the synthetic biology. So as I suggest, there were some phenomena. So here are a couple of proteins that we adopted to understand the LOB protein. So these are some fast cycling kinetic.
2 more minutes.
Yeah. So where we make the bond, formation and breaking very fast, or it could be intermediate, or it could be slow. So now we understand the networking through these colors codes, where the different part of the protein talk to each other and also to the chromophore. But then how this protein environment actually compensate the molecule, that is phenomenal. So we can see, if we look at the bond length of the chromophore, we see kind of mix where major changes kind of happening here, and there are some mixed phenomena. But then when we look at the kinetic wise, we can see the first one, they have a significant alteration in the bond length, and that's because of the near environment. But as soon as we go to, say, intermediate or slow kinetics, where the time span of making this adduct is much higher, we see the bond length is 0.001 Armstrong, and that is very negligible. And so by understanding the protein environment, we can actually now construct a lot of synthetic proteins, which can pave role to the therapeutic applications. So with this, I would also like to take the opportunity to introduce my school. So this was established in 2021, and basically we are like a baby in the plethora of big institutions in the nation. So this is now named as Jyoti and Bhupat Mehta School of Health Sciences and Technology at IIT Guwahati. And this is my research group, and we are also lucky to have funded by some major funding bodies. And thank you very much for your time. And I'm now open to questions.
Thank you, Professor Kaur. An exciting topic, actually. This is the application of QMMM in biological systems. So now this, you can question him if you have any doubts, clear from him. Students.
I have one question. Can I ask?
Yeah,
Sure.
please.
Thank you for your talk. Nowadays, trend is to use entangled states. So my question, do you see that states like correlated, entangled, or squeezed could provide additional clarification or bring some new physics in your calculations?
Yeah, right. I mean, the entangled states are of real importance. So here, just one example where I showed like a lot of snapshots are actually important to, describe the whole phenomena, because the traditional method is to take one, and then, the overall system behavior will be depicted just by one toy model. But rather than that, if we look at the dynamic insight and couple it with the quantum mechanical phenomena, so minor contribution for all the entangled states will be of high relevance. So definitely, I agree with that. And now, like unlike later days where we are only equipped with CPU computers, now a lot of the quantum mechanical algorithms are capable of running in the GPU system. So basically, we can make use of graphical processing units to speed up the calculations, and now it is quite possible to take account of those states.
So another question, since you are talking about the numerical algorithms, so what type of numerical algorithms do you use, and also the software packages?
Yeah, so typically the TD-DFT things, Gaussian and Orca are quite useful. And when we want to go to these wave function based methods like ADC2, CC2 kind of things, so we use TurboMole. And the one which I mentioned about the GPU computers. So now we have TerraChem, which is quite fast. And unlike the Gaussian software, which normally take the calculations much lengthier, it can do a bit faster with some approximations. Although there are some compromise, but then with this time effectiveness, we can compute a lot of things. So TerraChem is one of the major advancement that can help us in doing calculation in GPU. And besides that, QChem, I think, is also nowadays going to implement the same thing with GPU features. So the numerical and analytical kind of solutions, they are basically playing the key role.
Thank you.
Anyone? Any comments? So I have questions to Professor
Yes.
Paul. Very nice talk. I just wanted to know that, did you have ever compare with the implicit and explicit calculations?
Yes.
Like you say that PCM model, you can use it for TD-DFT, and you are saying that you are using the environment with the explicit interaction. So have you compared with the results anytime? Because I'm not sure,
definitely.
because I don't know how many such, suppose you are doing an environment like water molecules. So how many water molecules they're going to keep it to have an effect with the PCM model. So have you done it with the small calculations?
Yeah, we have done. I mean, you rightly pointed out, like we do not have a limit in that case. So
Yes.
I showed one example where we had a different kind of quantum mechanical. Yeah. So where we have this, for example, we completely use the implicit model, and we just mentioned, say, for this is for water. If you use 40, it will be chloroform, 4 for DMSO, like this. But then we can also place the water molecules. And now the question is the choice of placement. Because say, for example, when we take a polar molecule like water, we know that it will make some bonding here. And it is very unlikely to make some bonding in the hydrophobic regions. So ideally, we will place some water molecules here, and it could be 4 or 5, so that there should be a bonding chain link present. And this gives a biasness, actually. So the more water molecules we add, it could be reliable, but then it will be costly also. Again, the placement will play a major role. So there is no such limit. And
depending on the computational facility that you have, and what is the basis set that you are working on. Okay.
Exactly.
So mostly that TerraChem that you mentioning, is it based on the QMM methods?
Yeah, so it is taking part for QMMM. So we can do ground state calculations and also excited state calculations, and speed is pretty fast. So for example, for my system, not only this, but I also accounted for say 70 atom or 200 atom system, and I was able to reach say 15 picosecond in a single day. So it was really huge. And earlier it was paid. So I think in a couple of months, it will be free version also, which will be Python based. And so like everyone can actually use.
So mostly you are working on the ground state and as well as the excited energy gap, particle excitation that mostly you are going to see, right? So in general.
we also do this. For example, where I showed the
interaction
free energy.
energy.
Yeah. interaction, and then we do some designing. And now we are also using this flavine with some materials. So after joining, I started with material parts like graphene oxide and some other heavy metal doped system, and we are working with biological systems. So it is very fascinating to see how the quantum mechanics can actually reproduce the experimental measurement, and it makes life much easier to explain them.
Thank you so much, Professor Kaur, for an enlightening talk. Yeah, so over to the organizers, if you have any other comments, because I think we are done with this session.
Can I ask one more question?
Yeah,
Sorry.
yeah, please, Ranjit, please.
Why do you say when we need to solve time-dependent Hamiltonian problems, it is like a black box?
Yeah, absolutely. I mean, that is a fantastic question. So this was for students mostly. So as I said, like the typical method like TD-DFT, where we want to say calculate, and we can definitely use B3 leap, we can definitely use Cambridge leap or Omega 97 or B leap, whatever we want. But the question is how we are going to use. Many of us are not aware. So we simply write the keywords of the functionals and the basis set, and we rely on the output. And there, the orbitals that are involved in transition, the first excited state, second excited state, and so on and so forth, are not in our control. On the other hand, if we are well aware that one particular lone pair is not at all involved, then we can actually skip it out, and we can confine to remaining orbitals for linear combination of these atomic orbitals things, which will now give us much more accurate electronic transition energies. But then again, the thing is, in order to go for this active space things, we need to be 100% assured with our chemical intuitions that what orbitals we are using are actually important. So, so for traditional DFT methods, it is of course the black box method, because we are just about to use the functional names rather than higher method like CASPT2, NEBPT2 or CAS-SCF. We are in charge of the orbitals that are being involved in the calculations.
Thank you.
Yeah, welcome.
Okay, thank you all. I think thank you, Professor Kaur, for the nice talk, and over to the organizer. Yeah, thank you.
Thank you very much, Professor Das and Professor Singh. Thank you for the questions, and I really like talking on this platform.
Okay, thank you very much. Yeah. Let us actually thank both the speakers today in the morning today, and I just like to tell the organizers that I'm done. Yeah, if they have any other announcement, they can carry on. Thank you.
So thank you all for the being present here, and our next session, evening session, will be starting from the 3 p.m. Okay.
Thank you.
Thank you. Bye.